Today I'm going to talk about agar gel precipitation test, or also called as agar gel immunodiffusion test. You can use petri plates, glass slides, even small round plates can be used, and other materials that would be required will be the gel punch that I have made out of this plastic pasture pipette or from the PCR tube, and you may require needle to take out the gel. These are pipetters, automatic pipetters and mechanical pipetters. This is a pipette that you require, micropipette and micropipette tips. Agarose will be required, PBS to make agarose suspension or agarose solution, and of course you will require different samples including positive control, negative control, antigen and your serum sample, test samples. You require balance for weighing the agarose. So here I'm going to weigh the agarose to make 1% suspension. I am weighing 500 milligram of agarose, and so this is now put it into the flask. We'll measure 50 ml PBS since we are going to make 1% agarose, so 500 milligram in 50 ml PBS will make 1% agarose. So you can use 0.5 to 1.3, 1.4% agarose to carry out immunodiffusion test or agarose gel precipitation test. Swirl it and put it on the hot plate. Let it heat it up, and once it is molten, then it can be used to pour. Meanwhile, you can draw the templates depending upon which format you are going to use. For example, if you're using slide, then make a template for the slide. If you're using larger petri plate, you can use you have to draw the template for the petri plate. So taking a blank paper, you can draw their shapes. Similarly for the small petri plate or small cell culture plates, you can use this. Then draw the points where you want to punch the wells in the agarose. So these wells can be approximately 0.8 mm to 1 centimeter apart. They should be equidistant. So you have a central well, and then you have peripheral wells. In the central well, you will place the known reagent, for example known antigen or known antibody, and in the peripheral wells you'll have different types of samples, maybe your positive control, negative control, and test samples. So you can see different ways to create wells. So we have created the template, and now using the mechanical pipetter or with the rubber teat or with the automatic pipetter, we will take the molten agarose, which has been cooled down to 55 to 60 degrees centigrade, and it is now ready to be loaded into the petri plate. So we are adding around 12 to 14 ml of agarose into this 90 mm petri plate. Enough agarose should be added so that you can punch wells into the agarose. Onto the slide, around 4 to 5 ml will be enough. So if you want to carry out your assay on the slide, then this is how you do it. You make sure that you do not overload, and then once they are solidified, you can start to punch the wells with the help of this dropper, plastic dropper that had been cut from the front. You just push the air and press gently into the agarose and let the air in by when the air comes in, the agarose punch will also come in with it. Similarly, you can do it with the Petri. So push a little air out from the punch and insert it gently to pierce the agarose. Release the pressure so as to let the air in into the gel punch. This will suck in the agarose plug into the gel punch or plastic dropper. Repeat this for all wells. And now we need to seal the bottoms of the well. For this you can use a little bit of molten agarose to seal the wells. If you don't seal the well, then the reagents will leak through the unsealed wells and will be lost. So it is important to seal all the wells. So I'm sealing all the wells. And you can check the wells if the bottom have been sealed properly or not by holding plates or slides in your hand and tilting them so as to see a convex mirror-like shape or reflection from the bottom of the well. You may notice that at 12 o'clock the well is not properly sealed. You would like to seal it again. Yeah. Once all required wells are sealed, then we can add the immunoreagents or samples. For this, mark the wells on the back of the slide or the plate and add reagents accordingly. So here I have marked negative for negative control, positive for positive sera control, S for the sample, test sample, and the central well is where the known antigen will be added. So you can add around 10 to 12 microliter or 15 microliter soluble antigen in the central well, and equal amounts of reagents should be added in the peripheral wells. In the same manner, we can apply reagents to glass slide. I am placing antigen into the central well and test serum sample and positive serum samples into the outer wells. So this is a test serum sample that we are testing, and on to the left you can add the positive control, but make sure wells are marked so that no confusion is there while reading the results. Here I have not marked the wells just to show you, so you can mark the wells before. So this is how you can mark at least positive control, so you know which side is your sample. It is important to, and once you have done this, place this slide in a humid Petri plate, and then these can be incubated for 24 to 48 hours at room temperature or even at 37 degrees centigrade in the incubator. Humid chamber is required so that the agarose does not dry up and interpretation. After incubation, we'll look for precipitation lines between antigen well and positive control and test samples. So look at this figure. You see there are zone of antibody excess and zone of antigen excess and zone of equivalence. It is this zone of equivalence where maximum interaction takes place. At this particular concentration of antibody and antigen, large complexes will be found between the antigen and antibody. They become insoluble, and when they become so large and insoluble, they are not able to move, and they can precipitate in the form of a precipitation line that we have to look for between these wells. So you can see some real images and very well appreciate the precipitation lines between central well and peripheral wells depending upon different types of interaction between antigen epitopes and paratopes. We may be interested in the interaction between antibody and different antigens, or we may have a known antibody and want to test for or detect the antigen against the antibody. In that case, antibody can go into the central well and samples containing antigen are placed in peripheral wells with proper controls. For more detail, you can have a look at in your manual. And thank you for watching.